Hey everybody, we're going to start tackling respiration today. Probably divide into two parts. It's pretty long, so let me get this pulled up here. And here we go. So for cellular respiration, we're going to look at how things are going to do this. How do they process energy? The energy is stored, right? We talked about that. Energy is stored in the organic molecules, carbohydrates, fats, proteins, and heterotrophs eat these organic molecules, which is our food, right? And then we digest them, we break them down, and we get fuel out of this, right? And so this is a control, what we call a controlled release of energy, um, burning the fuels in a series of step-by-step enzyme-controlled reactions. This is very important, actually, and I'll show you why that is in a video. Um, so if we look at the two major things that we're going to talk about the rest of this unit, which is respiration and photosynthesis, you should probably by now, have been taught that there is a reciprocal relationship between the two. So the products of one are the reactants of the other, right? So they rely on each other heavily. It's really this is the engine that runs the planet, right? If this system broke down, we really wouldn't be able to sustain life anymore on our planet. So pretty important. So the equations are down here at the bottom. So get familiar with those. I'm not necessarily going to ask you to memorize the equations, but you should know the inputs and outputs, and kind of, why they're there and where they come from. And we'll be going through a lot of that as we go through here. So metabolic reactions are tightly controlled. So the energy isn't all released at once. And like I said earlier, it's very important. There's a video I'm going to post um, titled Rest in Peep. And so our little friend here on the page should give you a clue as to what might be in that video. But if you want to see why you want to have your energy released in very tightly controlled reactions, check that out and see what happens with just one of these peeps. So how do we harvest energy from fuels, right? So we break bonds and move electrons from one to another. And if you want to know where electrons are moving, follow the hydrogens, okay, at least in equations. Um, if you look at this box here, and yes, we're going to talk about some redox reactions, so you might have some nightmares from your chemistry days. So when something loses an electron or gains an electron, right, we're going to pay attention to that. And so you can see on the first side, if it loses the electron, the yellow one loses, the blue one gains. And then on the other side, because the yellow one lost an electron, it lost a negative electron, it became positive. And we call that oxidized. And the blue one gained the electron. It gained a negative, and so we call that reduced, right? Because it gained a negative, we say it was reduced, and that's where the hydrogen went. So if you want to follow where the electrons are going, follow the hydrogens. Okay. A little bit more on redox here. It shows you in the equation, the respiration equation here, what's actually being oxidized and what's being reduced. So again, follow the hydrogens. So this should work. Follow the hydrogen. So here's the sugar, right? Here's the glucose. And so this one became oxidized because look at all these H's, and you go over here and they're not there anymore. The H's are gone, right? The H's are now over here. So the oxygen that we take in gets reduced by taking up the hydrogens and making water. Okay, and then the sugar that we break down gets oxidized and becomes carbon dioxide, which we will then blast out of our mouths for the plants and help that process keep going. Okay, so glucose will be oxidized, oxygen will be reduced. Okay, and so respiration is catabolic and exergonic. We just talked about that in the other set of notes. So here's also another little table that may help you with oxidation and reduction and how to know what's being oxidized and what's being reduced. So if you're being oxidized, that's adding oxygen, removing hydrogen, loss of electrons, releases energy, and exergonic. And then over there on the reduction side, removing oxygen, adding hydrogen, gaining electrons, storing energy, and that is endergonic. And maybe, if I get my head out of the way here, maybe you guys were taught this little mnemonic of oil rig. Oxidation is loss of electron, and reduction is gain of electron. Okay, so I was always taught oil rig all the way back to my college days, I believe, or high school days, and so that's one of the things that like literally stuck with me since way back when. So use oil rig if it makes sense for you. So how do electrons move around? Well, we actually have electron shuttles. Okay, so we are going to have we kind of call them NAD and FAD. You'll hear them referred to as NAD and FAD, or NAD, NADH, and FADH. If it has the H, that's the reduced form, right? Because it has the hydrogen. If it doesn't, that's the oxidized form. But you'll see NAD plus and NADH, and then FAD and FADH. Okay, so these are storing electrons from our food. They exist in both forms. Um, the electrons come with protons, so that's why, right? Hydrogen, if you think about it, has one proton, one electron, and that's how you know you can follow if the hydrogen moves, it's taking an electron with it. And so this is how we're going to move our electrons around the cell into different molecules and to make things happen. So we will see a lot of NADH and FADH as we go. So the overview of cellular respiration. So I'm already regretting that I didn't change something. I usually only refer to this as 3 stages instead of 4. I used to do 4, but I like 3 better now, and I keep forgetting to change that. Not a huge deal. I'm not going to ask you. It all happens. It's just one of them is so minor. It's kind of a transition step that it probably doesn't deserve its own number. So the first main part is glycolysis. That's going to happen in the cytoplasm out there. And so let me grab a pen here. So glycolysis is going to happen out here in the cytoplasm. And you can look at this diagram and see all the inputs and outputs and exactly what's happening. So here's glucose, and it's going to be converted to pyruvate through glycolysis. And the electrons that are generated, the electron carriers, NADH, is going to go all the way over here, and we're going to save those for later. Okay. And then we're also going to generate a little bit of ATP, and all that's going to happen in the cytosol. And so in aerobic respiration, okay, if it's aerobic, then we're going to use oxygen for sure, okay? And it's going to take place in the mitochondria. Pyruvate oxidation, that's our transition step. That actually occurs about right here where this arrow is. That's going to be this step right here. The Krebs cycle is going to take place inside that mitochondria matrix, and the electron transport chain is going to take place on the mitochondria cristae, or that inner membrane that's all curved up. Okay, and so you can check out the equation down there once again. So how do we do this? Okay, so what's eventually going to happen? This is one of the very last steps. What's eventually going to happen? This purple guy is going to become super important. He's called ATP synthase. So what do you think he does? He's an enzyme, right? He's an ACE, and he's going to synthesize ATP. That's the thing that's going to make our ATP for us, and it's going to happen because hydrogen's going to flow through it, and it's going to turn it. It's going to spin it. This is another video I will put up for you. Maybe not, today or tomorrow, but you'll see it during the unit where you'll see that thing work, and it'll make a lot more sense when you see this video. And then it's going to set up a gradient, and that gradient is going to be really what generates the possibility of making ATP. Yeah, I had a couple other things on here. So it's going to take an ADP and a phosphate, and then it's going to push them together and make ATP. But again, this is one of the last steps. I'm trying to show you where we're headed. Okay, this is where we're headed, but we have a lot of work to do before we get there. So again, keep moving my head today. So big idea. Living organisms obtain energy by breaking down organic molecules during cellular respiration. And the main reason is to harvest the electrons from those organic compounds and use that energy to make some ATP. Okay. I actually thought I went through here and tried to fix a lot of that, and apparently they're not all fixed. I don't know why it keeps messing up. You probably noticed on the last slideshow as well. I did go back and modify that one and repost it, so hopefully it's fixed now. There's something weird going on with my presentations right now, and it keeps resizing a lot of the bubbles, and so the letters are getting all messed up. So again, why are we after those organic molecules? Because the energy's in the bonds, right? We've said that since the first unit. So again, the equation, just trying to go back through and show you where everything's coming from. So we have sugar, right? The C6H12O6, that's glucose, that's sugar, comes from our food, okay? And then we have oxygen, which you are breathing in because the plants give it to us from their byproduct of photosynthesis. You're going to exhale your CO2, and you're going to exhale a little water vapor, and then whatever, water's not used by the cell anyway, we will exhale. Some of it is used by the cell. And then when we get our energy, we will get our ATP. And thank you, plants, for the oxygen. So here's that picture again. And honestly, you can almost learn everything you need to know from this diagram. It's a pretty important one. So a couple things you'll want to look for. Inputs and outputs. So what's going in and out of each process? Where is it occurring? Okay. And not every specific is on here, but pretty close. It tells you where, it tells you the inputs and outputs, it tells you what's being made, what's being produced, and how everything's kind of moving. Now, there's a lot of details that goes into this, so we're going to continue with some of that. But this really tells you a whole lot about what's happening and where it's happening. So glycolysis or glycolysis would be splitting sugar, okay? And so it's going to occur in the cytoplasm, and it is anaerobic. So we don't need oxygen to run glycolysis. Lots of things run glycolysis, and they're not all aerobic. So this process has remained anaerobic. And for glycolysis, for splitting sugar, we're going to take a glucose, which has 6 carbons, and we are going to cut it up into 2 3-carbon blocks. Okay, and those are going to be called pyruvates. Okay, and so in order to do this, in order to break down glucose, we're going to have to spend some ATP. They're not just going to break down on their own. We're going to have to get the ball rolling. Okay, so you have to make an investment. You have to spend a little ATP in order to get more out of the back end. Okay, so you spend 2, but you get 4 in return, and that 4 is just from glycolysis, not from the whole thing. Okay. So here's the actual glycolysis equation, which I will not mess with very much at all. I thought you should see it. This is also something I will not mess with. I just kind of like telling you and showing you all of the little steps that goes on. Like this is glycolysis. All of this really happens. We are going to boil it down into just a couple of very simple steps. But if you get into biochemistry in college and stuff like that, you're going to see a lot of diagrams like this. And so I kind of just like to share those with you. Glycolysis is the investment step. This is where we spend a little energy, a little ATP in order to get the ball rolling so that we can then get some more ATP out in the end. So we're going to spend, let me grab my pen. We're going to spend up here a couple ATP up here, and so we're going to end up with 2 ADP here. So where did the other phosphates go? Well, they went right here. We talked about this in notes the other day. Okay, things don't just happen. They had to be phosphorylated, right? So phosphates go in here and they destabilize that glucose, and now they start breaking up in G3P. They make a couple of G3Ps is what we call them. And there are things, these arrows right in here, there are things in here, okay, all the way in here. Those are a lot of those other steps that I showed you in that crazy diagram a second ago. There's actually about 10 reactions that happens in glycolysis. We're just going to kind of show you the beginning, how it goes, and then we're going to skip to the end, to the good part, because the College Board is not going to care that you know all of those steps. In fact, they don't really want you to. So by the end here, we're going to have a couple pyruvates, and we're going to make 4 ATP, but remember that's only 2 net gain because we spent 2. Okay, so we'll look at that when we get to the accounting. So here's our same thing. We also produced some NADH. We made some NADH, and notice those are electrons going in the piggy bank. Okay, electrons going in that NADH piggy bank because we're going to save them for later. Okay, we need to save those for a rainy day, and we're going to use them later in the process. Glycolysis summary, the energy investment. It's endergonic. We're putting energy in, invest some ATP. The energy payoff, it's exergonic. Harvest a little ATP and a little NADH. We're not getting much out of glycolysis, but it's a start. Okay, so what did we make? We made what I like to say is 2, 2, and 2. 2 pyruvate, 2 ATP, and 2 NADH. Okay. Whoop, there you go. 2, 2, and it's not listed there, but the 2 pyruvates are there as well, and those are going to carry us on into the next step. So substrate level phosphorylation. So there's a couple of different types of phosphorylation in this process. And so ATP is produced by enzymatic phosphate transfer from another organic phosphate. Okay, so in this case, glycolysis does what we call substrate level phosphorylation. So in this case, look, there's just a substrate here, and here's an enzyme, and they're just general. We're not giving them any specific names here. But it's going to peel, this enzyme is going to peel a phosphate off and add it to the ADP. And when you peel that phosphate off, that circle is now going to become a square. That's a different product now, because once you pull that phosphate off, it will rearrange, right? There'll be a conformational change. Now it's a different shape, and we made some ATP. Okay, so just know that the phosphate comes from another substrate during glycolysis, and there'll be a couple of substrate levels, and then there'll be another one toward the end. Evolutionary perspective, how long has this been going on, and how does it work? Well, prokaryotes were the first cells, right? No organelles, anaerobic atmosphere, so life was oxygen free for a while, and so energy had to be captured from organic molecules in the absence of O2. And prokaryotes evolved. Glycolysis are ancestors of all modern life. All cells, okay, all living cells, no matter what they are, still utilize glycolysis. So that means all of those enzymes that run glycolysis and all those steps I showed you, those enzymes are very well conserved. Okay, that's kind of a term we use when something has stayed the same for a very long time through evolution and they haven't changed much because they work really well and they've done their job, and it's just hasn't needed to change, I suppose. No mutation ended up being more beneficial so far. So again, little overview of glycolysis. You have your inputs, you have your glucose, you have your NAD plus, you have your 2 ATP, and on your outputs you get your 2, 2, and 2, your 2 pyruvate, your 2 NADH, and your 4 ATP. The electrons down, the electrons down there in the piggy bank are going to be saved for later. You can see that bar that leaves the glycolysis part here. So here is where we're going to use the NADH much later on down here in the electron transport chain. So what's the point? The point is to make ATP. That's what we're really trying to do, right? So we made a little. Did we make enough? Probably not to run the human body. So also you could run out of NAD. You have to have a way to regenerate more NAD. If you just made NAD into NADH, like pretty soon you'd run out and then energy production would stop. And so another molecule has to take that hydrogen, okay, from NADH so that NAD gets freed up for another round so that it can recycle and go back and pick up more, okay? So what happens next? Well, it kind of depends on what's happening. The fate of pyruvate depends on oxygen and whether it's available or not. So here's the branching point. So if there is no oxygen available, then pyruvate will go down a path that goes toward fermentation, okay? Anaerobic respiration, okay, would be fermentation. Okay, it happens in the cytoplasm, so it's the same place out there with glycolysis. If oxygen is available, then pyruvate's going to move into the mitochondria, and it's going to run the Krebs cycle, and it's going to go through aerobic respiration. Okay, so you can see in the diagram to the right as well, there's that branching point there. And there's a couple different types of fermentation that we'll get into here. There is alcohol fermentation, which is only done by bacteria and yeast. Okay, and if you look at this reaction, you have pyruvate, which is a 3C, and then it converts to ethanol, which is a 2C. And there's also some carbon dioxide, which is just the 1C. And that carbon dioxide leaves, right? Carbon dioxide is going to get released out from that reaction. And so what happens is this becomes a dead end process because when 3 goes to 2 and that third C that was the CO2 gets released, there's no other third C to go backwards. You cannot run this reaction backwards. It just goes one way. And so you can't reverse this can't reverse this reaction. And you can look at this funky diagram down here at the bottom and go through it if you like and count the carbons. Not really necessary for me, but if you'd like to, have at it. And then if we looked at lactic acid fermentation, we have animals and fungi that do this. So in this case, pyruvate turns into lactic acid, also a 3 carbon. So do you think we can run this in reverse? Absolutely. Okay, so we can do that. And once O2 is available, once O2 becomes available, then we can run that process backwards. So if you're not sure, how your body or when your body needs lactic acid, here's an example for you. If you work out really hard and you've been, let's say you haven't been working out and you try to go for like a 5-mile run, probably not going to go great for you. You're going to just be consuming oxygen, right? Trying to work so hard, and your body is going to try to keep up and give you all that energy, but your cells are probably going to run out of oxygen at some point, or it'll get very low. And so it'll start doing fermentation. It'll do lactic acid fermentation. And that next day, when you wake up and you're really sore, that's the lactic acid. Lactic acid builds up in your joints. And so that's why your muscles get so sore after you lift weights or run too hard or whatever. If you overdo it and you're sore, it's because you generated some lactic acid. But then once you recover and you sit on the couch and you watch some Netflix for a while, then your body is going to keep breathing in oxygen, and it's going to filter through your body and provide enough oxygen then to then run this reaction backwards from lactic acid to pyruvate, and then the pyruvate will go into the mitochondria and go down the other pathway and give you some more energy so you can get back out there and try again. Okay? So glycolysis is only the start. So we made a couple pyruvates. Pyruvate has more energy to yield. There are 3 more carbons there, right? We only split, a bond or 2 there to get these into 3 carbon blocks. And so we're going to keep stripping out all of those carbons and breaking down those bonds to get that energy out. So if O2 available, pyruvate's going to enter the mitochondria, and then the enzymes of the Krebs cycle will take over, and they will keep oxidizing the rest of that sugar. Okay? So here's our picture again. So we've completed glycolysis. We talked about fermentation as well. Our pyruvate is ready to go into the inside of the mitochondria, and that's going to be where that transition step takes place. So we've made a little ATP so far, but not near enough. We're going to have to make some more. So the Krebs cycle is going to take place in the mitochondria matrix, and again, there's our picture over there, and the matrix is the inside. The matrix is this space. Let's see if I can color in here. The matrix is all this space in here, okay? That's the matrix. And then the cristae is the actual membrane that is really curvy in there. That's the membrane in there. And so those are some parts that are going to happen with the Krebs cycle, and it is going to be an aerobic process. Okay, Hans Krebs won the Nobel Prize in 1953 for kind of sorting out the Krebs cycle. And give me 1 second. I need to pause for 1 minute. All right, I'm back. Sorry about that. So yeah, the Krebs cycle, also known as the citric acid cycle, because that is one of the components of the cycle. It's going to occur in the mitochondria matrix. It's going to occur in that space. It's an 8-step pathway, lots of enzymes involved. You don't need to know all of the steps. It evolved later than glycolysis. So does that make evolutionary sense? Well, this one's aerobic, right? So it probably evolved later than the anaerobic process. So bacteria, about 3.5 billion years ago, were doing glycolysis, and then planet was getting oxygenated, and then, several years later, many years later, you had free oxygen, and so photosynthesis started. Eukaryotes about 1.5 billion years ago, and then aerobic respiration with organelles, and then mitochondria started taking off. So that's just kind of a timeline for you to look and see how all of this stuff evolved. So this is what I mean by the transition step. So this is what was listed as like step 2 in there, but it's just kind of a little transition step to get pyruvate into the mitochondria. So here's your pyruvate, and it's going to go through this transport protein, okay? And then you have another enzyme here called coenzyme A, okay? And so when pyruvate comes in and it merges with coenzyme A, and there's a reaction that takes place, we get something called acetyl-CoA. Okay, so pyruvate gets converted to acetyl-CoA, acetyl coenzyme A. So remember, pyruvate was a 3-carbon. Acetyl-CoA is a 2-carbon. So we lost a carbon. Where did that other carbon go? Well, during this reaction, we had some CO2 there. And so it was a byproduct. And so you have that CO2 gas that's in there, and gosh darn it, I thought there was an arrow that was going to show you that it would leave, but that CO2 is going to leave, right? That's going to be part of what we exhale. And so the CO2 will get out of there, so we go from a 3-carbon to a 2-carbon acetyl-CoA. And then you have a little transfer, you get a little reduction there, and so there's some NAD that becomes NADH. And so you have a little bit of that as well. And you have the formula down here at the bottom. So you have 2 times the 2-carbon sugar plus the NADH plus the CO2. And it's 2 times because in glycolysis you generate 2 pyruvates. Okay. So here's another picture of the Krebs. So let's look at the words first on the left-hand side. So the Krebs cycle produces more electron carriers, more NADH and FADH, and those are going to head to the electron transport chain eventually, and you're going to get a little more ATP. Okay, so if you look, here's our pyruvate from glycolysis, and here's our acetyl-CoA. So that's the transition step. You can see up here. So you can see here's our CO2, here's our reduction, our NAD to NADH. Here's our acetyl-CoA, right? And so acetyl-CoA is a 2-carbon, and it's going to enter the cycle, and it's going to immediately join this 4-carbon compound, okay? And so the acetyl-CoA plus the 4-carbon is citric acid. This is why it was also called citric acid cycle. So now we have 6 carbons again, okay? The 2 from acetyl-CoA, the 4 here, we have 6 carbons. And it's going to go around this process here, and lots of things are going to happen. So you're going to have NADH or NAD get reduced again to NADH. You're going to give off some more CO2 right here. And so we're going to lose a carbon there. So we're going to go from 6 to 5. And now we're going to lose another CO2 off of the 5. So we lose another carbon, and we go from 5 to 4, which is the one all the way over here, with some intermediate steps in here and some reductions going on as we go. We made a little FADH here as well. And here's where we're making our ATP. So a lot of electron carriers. Look, the transition reaction has one. Here's electron carrier. Here's electron carrier. Here's electron carrier. Here's electron carrier. So it's a really important step for generating the electron carriers. We're just getting a little bit of ATP out of it, but that's how all of that goes. And now you have all of the electrons stripped from the molecules every time a carbon is removed. So we're breaking down that original glucose into, stripping it down and getting all of those electrons out of there so that we can then make some ATP. Okay. And the CO2 is going to go out, okay? That's what we're going to give off for plants, and we're going to save our NADH and our FADH2 in our piggy banks for the electron transport chain. So if you want to count the carbons, you can go, there's 6 at citric acid, there's 5 down here, and then there's 4 again right before it meets up with the 2 from acetyl-CoA to make 6 again, and then it'll go around again, okay? So CO2 is going to go out. We're going to make a little ATP. So we completely broke down glucose. Glucose got broken down from C6H12O6 to CO2, to carbon dioxide, right? And that's what we give off as a byproduct. And we only ended up with 4 ATP. So what was the point of all that? Only 4 ATP. Well, we need electrons. We need electrons for the electron transport chain. And so we still have that money in the piggy bank that we're going to use later on. So what happened in Krebs? Let's review and do some counting here. Pyruvate converted to acetyl-CoA. Acetyl-CoA goes through many reactions in the mitomatrix. produces 2 ATP, a bunch of NADH, some FADH2, so 282. So some of all of that, and we're going to give off some CO2, which the plants will thank us for. Okay. So a little bit of accounting. It's not quite as straightforward as glycolysis, actually. So don't worry too much about this. You just need to know the majors. Okay, so here's your net gains. Whoops, I'm sorry. Your 282, you got 2 ATP, you got 8 NADH, and you got 2 FADH. Okay, and so it was really a 414. I'm sorry, a 141. But then you have to times it by 2 because you had 2 pyruvates. So then you get 2A2. So what's the value of Krebs? If you're only getting 2 ATP, why go through all of that? Well, those electron carriers, that's going to be what makes the difference for us down the road. Okay, we broke down all the glucose, we stripped everything out of there, and now we have some reduced molecules that we can move over to the electron transport chain, and then we can really start making some ATP, hopefully. So what's the point? The point is to make ATP. So we've gone through glycolysis and Krebs. I'm going to stop the video here. I think this is probably long enough for now. And then I will make another one that will tackle the electron transport chain and a couple of other things that we'll clean up at the end. And it'll probably be, about 2/3, 1/3 or so. But anyway, here's the first part of respiration. So hopefully you guys are doing okay with it. And hopefully that was helpful for you. See ya.